Okay, so you send over some really interesting research on these macrophages,
Hmm.
and specifically
Yeah.
this subset called fibrosis-associated macrophages, or FAs.
Yeah, they're pretty fascinating cells.
Yeah, and what I thought was really interesting is that the scientists at Genentech
Yeah,
developed this AI model called SCimilarity to help them understand these cells better.
that's right.
So it's kind of like
Yeah.
a massive search engine for cells. We're talking a database of 22.7 million cells.
It is. It really is like a Google search for cells.
Crazy.
But instead of searching for websites, you're analyzing gene expression data to find similar cells,
Wow.
even across different tissues and diseases.
Okay, so the paper we're looking at is called Scalable Querying of Human Cell Atlases via a Foundational Model Reveals Commonalities Across Fibrosis-Associated Macrophages.
Yeah, it's a mouthful.
It is a mouthful, but it does get to the heart of what we're talking about,
It does, yeah.
using this AI to understand these FLs.
Exactly. And I think what's really cool is that it's helping researchers uncover these connections
Yeah.
between FAs that we might not have been able to see before.
So let's talk about how this similarity thing actually works.
Okay.
It uses something called metric learning, which sounds very complicated.
It does sound
Can
complicated.
you break that down for us a little bit?
Yeah, so think of it like this.
Okay.
Imagine you're teaching a computer to recognize different breeds of dogs.
Okay.
You know, you show it pictures of golden retrievers and poodles and bulldogs,
Uh-huh.
and it learns to distinguish them based on their unique features.
Okay.
Well, Similarity does the same thing with cells.
Gotcha.
But instead of looking at, physical characteristics,
Right.
it's analyzing gene expression data
Okay.
to identify different cell types.
So basically learning to recognize patterns in the genes that are turned on or off in different cells.
Exactly. That's a great way to put it. And by comparing millions of cells, Similarity learns which gene expression patterns are important for distinguishing different types of cells.
So then how do they build this database of 22.7 million cells?
Yeah, that's a big question.
This seems like a logistical nightmare.
It's a massive undertaking for sure.
Yeah.
But they use a combination of automation and manual curation.
Okay.
So they created this pipeline to automatically download and process data that's publicly available
Huh.
from resources like the Gene Expression Omnibus.
I see.
It's kind of like scraping the internet for cell data, but on a massive scale.
Wow. I can only imagine the amount of data processing.
It's a lot. It's a lot of data.
Yeah.
And then they had to make sure that they were comparing apples to apples.
Right.
So they used something called the cell ontology, which is this standardized vocabulary for describing cell types.
Yeah, makes sense. You need to be speaking the same language.
You do.
But it sounds very time-consuming.
It is time-consuming up front, but the benefit is that once it's trained on this massive data set, it can analyze new cells incredibly quickly.
Oh, okay.
We're talking milliseconds for searches and seconds for annotation.
Wow. So it's like you're building this super highway of data,
Yeah, that's a great analogy.
and then you can just zoom along.
Yeah, once the road is built, you're good to go.
Okay, so we've got our AI cell matchmaker,
Uh-huh.
and we have this massive library of cells.
Right.
So let's talk about the stars of the show, the ephemedes.
Okay.
What makes these cells so interesting?
So, as their name suggests, they're associated with fibrosis,
Oh, okay.
which is basically the formation of scar tissue.
Right, and that can happen in any organ.
Yeah, it can happen in various organs. Lungs, liver, even the heart.
And too much scar tissue is bad news, right?
It is. It can interfere with how organs function.
Okay.
And so FMs have been popping up in a lot of fibrosis studies.
Uh-huh.
But here's the thing. Scientists have been using different names and marker genes to describe them,
Right.
which has created some confusion in the field.
Like, we're not even sure if they're talking about the same cell.
Exactly. Yeah, it's like trying to figure out if someone's talking about the same person
Yeah.
based on, like, vague descriptions.
And that's where similarity comes in.
That's exactly where it comes in.
Yeah.
It can cut through that confusion and say, okay, are these cells from these different studies actually the same?
Gotcha. So what did it uncover about FMs?
All right.
Was it able to connect the dots between these studies?
It was. It found that FNs are most prevalent in idiopathic pulmonary fibrosis,
Okay.
which is IPF for short.
Okay.
That's a serious lung disease.
Okay.
They're also abundant in systemic sclerosis,
Uh-huh.
which is another condition that involves fibrosis in multiple organs.
So that's kind of confirming what researchers already suspected.
Yeah.
But I'm sure there were some surprises, right?
Oh, absolutely.
Exactly.
So Similarity found FM-like cells in COVID-19 lungs.
Oh,
in various cancers like pancreatic cancer and colon cancer, and even in healthy lungs, although in much lower numbers.
hold on. In healthy lungs.
Yeah.
So these aren't always bad guys.
That's what's so fascinating about them.
Right.
They seem to be involved in normal tissue repair and maintenance,
I see.
but they can become problematic when things go wrong, like in the case of chronic inflammation or injury.
So it's like they're like a construction crew in your body.
I like that.
They're there to fix things.
Uh-huh.
But if they get out of control, then they start building where they shouldn't.
Yeah, that's a great analogy.
And so similarity doesn't just find these cells,
Right.
but it helps to explain why they're similar.
Exactly.
So it uses a technique called integrated gradients to identify which genes are most important for distinguishing FMs from other macrophages.
Yeah, that's a really cool feature.
So it can actually point to the genes that make these unique.
It can, and a lot of those genes are involved in processes that are related to fibrosis, like extracellular matrix remodeling,
Yeah.
which is basically the scaffolding that holds tissues together.
Others are involved in lipid metabolism
Yeah.
and recognizing damage signals,
Which makes sense.
which makes sense given their role in tissue repair.
Okay, so we're starting to understand where these FMs hang out
Yeah.
and what they're doing.
Right, and that knowledge is super valuable for researchers who are studying these diseases
Make sense?
because it could lead to new therapies that target FMs specifically
Right.
to hopefully prevent or even reverse fibrosis.
So studying cells in their natural environment, in vivo, can be really tricky.
It can be, yeah.
Wouldn't it be easier to study them in vitro, like in a lab setting?
Yeah, you're reading my mind because that's actually one of the next big things that Similarity helped to uncover.
Okay.
Remember how I said it can search through millions of cells?
Yeah.
Well, the researchers decided to put that to the test.
Okay.
They asked, can we find an in vitro model that replicates these FMs?
Uh-huh.
And guess what?
Did they find a match?
They did.
What was it?
And get this.
It was in a study that wasn't even focused on macrophages.
Oh, really?
Yeah.
Interesting.
The researchers were studying hematopoietic stem cells, or HSCs,
Okay.
which are the cells that give rise to all blood cells.
Right.
And they were growing these cells in a 3D hydrogel.
Okay.
It's kind of like a jelly-like substance that mimics the environment of tissues.
That's so wild. How did they even connect that to FMs?
It's all thanks to similarity.
Okay.
They found that when they cultured peripheral blood mononuclear cells, or PBMCs, in this 3D hydrogel,
Uh-huh.
a significant portion of them started to look and behave like FMs.
So PBMCs are just a mix of immune cells that are found in our blood, right?
Right, that's right. They're easy to obtain.
Yeah.
And they're readily available.
So they kind of stumbled on this way to create FMs while studying something completely different.
It really is a remarkable example of serendipity in science.
Yeah, what are the chances?
And it opens up so many possibilities for research.
So instead of having to extract these cells from tissues, which can be hard to reach,
Now we can just create them in a dish.
potentially, yeah.
Wow.
It means researchers can more easily study these cells, manipulate them,
Yeah.
test potential drugs on them,
That's incredible.
and learn a lot more about their role in fibrosis.
And they wouldn't have found this without similarity
Right.
and its ability to search through this massive data set.
It really highlights the power of AI
Yeah.
and big data in accelerating scientific discovery.
Okay, this is a lot of information. I think we need to take a little breather here.
Sounds good to me.
All right, so we've gone from this complex research paper to, I think, a pretty good understanding of FMs, their role in disease,
Right.
and the power of AI to uncover all these connections. But I have a feeling we've only just scratched the surface.
Yeah, there's a lot more to explore.
I'm ready for more.
Let's do it.
What other insights did Similarity reveal?
Okay, so one of the things that Similarity can actually be used for is to annotate cells
Okay, you mean like giving them a label? Yeah, giving them a proper label.
based on their gene expression profile.
Exactly.
So it's comparing it to that huge database that it already has
It is, yeah.
of cells that have already been carefully annotated.
Right, and remember, they use the cell ontology
Yeah, to make sure everything's standardized.
to keep everything standardized.
Oh.
So when you give similarity data about a new cell,
Yeah.
it compares it to this database and finds the closest matches.
Oh.
It's like finding its cellular family, you
Right.
know?
It's not just finding similar cells, it's actually classifying them
That's right.
based on those similarities.
It's classifying them at the single cell level,
Okay.
which is really important because a lot of other methods rely on clustering cells first.
Uh-huh.
which can be kind of messy and subjective.
Yeah, you have to decide how many clusters to create
Right, exactly.
and which cells belong in each cluster.
Exactly.
But with similarity.
With similarity, each cell gets its own personalized annotation
Wow.
based on its unique gene expression profile.
Okay, and you said this is really fast.
It's incredibly fast. It can annotate a whole data set of thousands of cells in a few seconds.
Okay, that's impressive.
It's pretty amazing.
But how do we know if it's actually accurate?
That's a great question.
Like, was it able to accurately annotate cells from data sets it had never seen before?
So to test that,
Yeah.
they used 14 different data sets
Okay.
that were completely separate from the data that was used to train similarity.
Right.
And these data sets covered a wide range of tissues and cell types.
Okay.
So it was a pretty rigorous evaluation.
Sounds like it.
And what they found
Yeah.
was that over 70% of the cell populations
Okay.
had high agreement between similarities annotations
Uh-huh.
and the original annotations that were provided by the researchers.
That's pretty good.
Yeah, it's really impressive considering that Similarity was working with completely new data.
And in some cases, similarities annotations were actually more accurate.
That's right. There were a few cases where Similarity actually outperformed the human experts.
Give me an example.
Okay, so in one kidney data set,
Uh-huh.
Similarity identified a group of cells as CD8 plus T cells,
Okay.
which are a type of immune cell.
Right.
The original researchers had labeled these cells as CD4 plus T cells.
Okay, so different kind of T cell.
A different kind of T cell, yeah.
So similarity was disagreeing with the human experts.
It was, and when they looked closer at the gene expression profiles of those cells, they
Yeah.
found that Similarity was right.
Oh, wow.
The cells actually expressed the markers for CD8 plus T cells,
Even though they had been mislabeled.
even though they had been mislabeled initially.
Wow, so the AI caught a mistake
It did.
that the human researchers had missed.
It's pretty remarkable to see AI advancing to the point where it can outperform humans in certain tasks.
Yeah, that is wild.
It's pretty cool.
Okay, so we know similarity can accurately annotate cells, even from data sets it's never seen before.
Right.
What else can it do?
Well, remember how we talked about that whole Google search idea? That's where it really shines. It takes that concept to a whole new level.
Okay, walk me through it.
All right, so let's go back to the FMs.
Okay.
The researchers wanted to use Similarity to search for FM-like cells
Makes sense.
across that entire 22.7 million cell database.
Yeah, looking for those cellular cousins
Exact.
across different tissues and diseases.
And the amazing thing is that it took less than 2 seconds
Wow.
for Similarity to find the top 10,000 most similar cells.
That's insane. 2 seconds to compare one cell to millions of others.
It's the power of metric learning.
Yeah.
And the results were fascinating.
Likewise.
So it confirmed what we already talked about.
Yeah.
That FMs are particularly abundant in lung samples from patients with ILD and systemic sclerosis.
No.
But it also revealed their presence in several other fibrotic diseases and tissues,
Okay.
like the pancreatic cancer and the liver metastasis we talked about.
Right. And even in healthy lung tissue, although in smaller numbers.
Exact.
Maybe they're not always bad guys.
Right. They might actually play a role in normal tissue maintenance.
Okay, so we're starting to see a bigger picture
We are.
of what these cells might be doing.
Yeah, but I think the most remarkable thing is that Similarity helped the researchers identify that in vitro model for studying FM.
The PBMCs in the 3D hydrogel.
That's the one. Remember, that study wasn't even focused on macrophages.
It was about HSCs.
Exactly. They weren't looking for FMs at all.
Right.
But Similarity was able to find them anyway.
Just by searching through that massive database
Just by searching through that massive data.
and identifying a match based on gene expression.
It's like the cells found themselves.
Yeah, that's crazy.
It really is.
So it's not just identifying similar cells across different studies,
Right.
but it can uncover connections between research projects
That's right.
that seem completely unrelated.
It's like a scientific matchmaker,
Yeah,
connecting researchers who might not even realize they're working on similar problems.
that's wild.
It's really cool, and it's already leading to new collaborations and breakthroughs.
Wow.
And, Similarity is being used by researchers all over the world
I bet.
to explore the human cell atlas, uncover new cell types, identify potential drug targets.
Okay, so what are they working on to improve similarity going forward?
Well, one thing they're focused on is incorporating even more data into the system.
Okay,
Right now, the database is primarily made up of data from adult tissues.
I see.
So it might not be as accurate when analyzing cells from, say, fetal or pediatric samples.
Right.
And they're also working on adding data from cancer cells and cell lines,
Oh,
which
right.
weren't included in the initial training set.
Makes sense. Those can be particularly tricky to classify.
They can be very tricky to classify.
Yeah.
And they're also exploring ways to integrate data from other species,
Oh, interesting.
which could open up even more possibilities for research.
It sounds like there are so many exciting directions for this technology to go in.
There really are.
Yeah.
It's just the beginning.
I'm curious to see what kind of impact it has on biology over the next few years.
Me too. It's a really exciting time to be in the field.
It really is.
And remember, this is just one example of how AI is changing the way we do science.
It really highlights the power of using these computational approaches
Yeah.
to analyze large data sets and uncover hidden patterns.
It's incredible what we can do now.
It is.
Okay, I think that's a good place to stop for now. We've covered a lot of ground in this deep dive, from the basics of similarity to its incredible potential for accelerating scientific discovery.
Yeah, it feels like we've taken a whirlwind tour of the human cell atlas.
That's a great way to put it.
Okay.
But before we move on, I'd
Yeah.
love to hear your thoughts. What stood out to you the most from what we've discussed so far?
So I think, honestly, it's hard to pick just one thing.
Yeah,
The scale of the database is just mind-boggling to me,
it is massive.
that we can compare and analyze millions of cells
Right.
so fast.
Yeah.
It's really amazing.
It's a game changer.
But I think what's even more exciting is the potential for this to translate
Absolutely.
into real-world benefits
Yes.
for patients.
That's the ultimate goal, right?
Yeah, so in terms of like developing new treatments for diseases like fibrosis.
Exactly. Being able to identify and study specific cell types like these FEs
Mm-hmm.
in such detail, it just opens up all sorts of possibilities for developing targeted therapies.
So instead of treating symptoms,
Right.
you can actually go after the cells that are causing the problem.
That's the idea. And because Similarity can identify similar cell types across different diseases,
Right.
it could lead to the development of drugs that are effective for a whole range of conditions.
So a drug that works for IPF
Mm-hmm.
could also be effective for systemic sclerosis
Potentially, yeah.
or even certain types of cancer.
It's possible, and the fact that they were able to identify an in vitro model for FEs
Yeah.
using Similarity
Right.
is huge for drug development.
Because it means we can test potential drugs in a lab without having to rely on animal models
Exactly.
or human trials early on.
It could really accelerate the drug discovery process
Yeah.
and hopefully lead to new treatments for these fibrotic diseases
Much faster.
much faster.
So it all started with this question.
Right.
Can we find an in vitro model
Mm-hmm.
for FNs
And Similarity found the answer.
hidden in this massive data set?
Hidden in a massive data set from a totally unrelated study.
Yeah, it's amazing.
It's really cool.
So it's like this detective
I love that analogy.
working behind the scenes,
Yeah.
piecing together clues from millions of data points.
Yeah, to solve the mysteries of disease.
Right, and we're still just scratching the surface
Absolutely. We're just at the beginning.
of what AI-powered tools can do.
You said they're working on expanding the database
Yeah.
and refining the algorithms. They're constantly working to improve it. And
Yeah.
as the technology continues to evolve,
Uh-huh.
we can expect even more groundbreaking discoveries in the future.
It's an exciting time to be in biology.
It really is. With tools like Similarity, who knows what we'll be able to uncover.
Well, I think this has given us a lot to think about.
I think so too.
We learned about this powerful new AI tool
Yeah,
that's changing the face of research.
it is.
And we've explored the world of FMAs
those fascinating cells.
and their role in disease.
Yeah, and how this research is leading to new treatments
Right.
and a deeper understanding of the human body.
So to our listeners, if you want to learn more about Similarity and the research that we talked about today,
Uh-huh.
be sure to check out the links in the show notes.
There's a whole world of cellular secrets waiting to be uncovered.
Who knows, maybe you'll be the one to make the next big discovery.
You never know.
Until next time, keep exploring.